Now, there are some processes on the apical pole of epithelial cells. However, these processes are some optional structures. I mean, some epithelial cells have them and some don't. It's some kind of an accessory feature. There are three different types of these things, and we're going to check them out on these three cells. The first type is like this. See, these are some folds on the surface. These folds are really cool because they increase the surface of the cell. You see, without folds, you have just this much surface. But with folds, if you open them, you will actually have this much surface. But why do we need a lot of surface? To absorb more stuff. With an unfolded surface, this cell can only absorb this much. But with the more surface which is provided by these folds, the cell can absorb this much. So you can see how this can come in handy, huh? By the way, we don't actually just call them folds. These processes are called microvilli. There are also some other processes on some other epithelial cells that are much larger than microvilli. These processes are like this and are called cilia. As you can see, cilia are much larger than the microvilli. And they also have a whole other function. While microvilli is made for absorbing stuff, cilia doesn't really care much about absorbing. It's more like a strong hand for the cell for throwing stuff. So far, we've got microvilli, which is small and helps the cell absorb stuff better, and then cilia, which acts as a strong hand that can push the stuff away. Also, it's really big. But there's another type in the middle, which isn't as big as cilia but also not as small as microvilli. We call this type of processes as stereocilia. Even its function is in the middle. It provides a little bit of good absorbing ability as well as the ability of pushing the stuff around. Also, its inner skeleton looks more like microvilli than cilia. So let's check these three once again. Microvilli are the small processes on some epithelial cells which increase the surface of the cell membrane. This provides the ability of a better absorbing for the cell. Cilia, on the other hand, are some very large processes that, unlike microvilli, don't have a lot to do with the absorbing stuff. But they can be strong hands for the cell to push the stuff around. And stereocilia are some structures in between, not too big, not too small, and they do both of the things these two other ones do.